All right, next we have Martina Zivna. And Martina holds a PhD in molecular biology, genetics, and virology, and is currently a senior scientist with the clinical research team led by Professor Stan Kamak, we had just heard, at the Department of Pediatrics and Inherited Metabolic Disorders at Charles University in Prague, Czech Republic. She participates in the international multi-site project aimed at identification and characterization of genetic factors contributing to inherited tubulostitial kidney disease, including REN, SEC61A, UMOT, MUC1, ALG5, and APOA4. She recently developed a non-invasive urine-based immunolabeling technique for diagnosing MUC1. And last year, we are just so proud and excited for Dr. Zivna. She was awarded a prestigious 4-year grant by the Czech Minister of Health to continue her research in the field of inherited tubulointerstitial kidney diseases. With that, thank you for being here, Martina, and I'll turn it over to you.
So, hello. I will share my screen. Can you see my screen? Can you see my screen?
Yep.
Yeah. Okay. So, good morning for US. Good afternoon for Europeans. So first, I would like to thanks to organizers to give me the opportunity to present in this wonderful conference topic about diagnostic strategies for rare kidney diseases. I joined the team, the Prague kidney disease team in 2002 as a PhD student when I joined the team of the Stan Kamak. And we are focused on identification and characterization of genetic causes of rare kidney diseases. In these days, we are clinical research team together with the team of Dr. Blair and Kendra Kidd, and we also worked very closely with other nephrologists and groups such as Irish group of Peter Conlon. So how we are working? Based on medical history and symptoms, we are collecting set of patients and families with a specific disease. First, we analyzed DNA to identify the causal gene and mutation for the disease. If we identify this mutation, then we can make a genetic diagnosis. And additional to the genetic diagnosis, we also aim to identification and characterization of the biological consequences of mutations, causal mutations, and the knowledge of genetic diagnosis and molecular mechanisms of the disease is basis for the therapy development. The universal biological concept proposes that genetic information encoded in DNA is translated to protein through transcription to RNA. Protein synthesis occurs within cell, and it's responsible for physiological functions of the cell organs and entire organism. In a human body or human DNA, there is around 20,000 genes coding proteins, but it's only around 2% of entire DNA molecule. And in health, all of these cascade is fully functional. If some of gene is mutated, it may lead to production of the protein that is ill functional. And such a protein may either not be included in the physiological functions and cascades, or may be toxic for cells that produce it. The disease type and severity depends on the type of cells that produce mutated protein or physiological functions that is responsible the affected protein. Now let me present hypothetical cases that we are solving within as a part of our genetic kidney research. So you can see different type of inheritance and clinical and biochemical presentations of the typical signs from de novo development of disease through recessive type of inheritance to dominant type of inheritance. And in such some of cases, the medical history or family history and clinical signs of the disease are not informative, and the genetic disease or genetic nature of the disease is questionable. In these cases, we consider genetic disease when the cases kidney fails or in individual kidney fails before 50 years of age. In ancient ages of genetics, the solving of cases was based on linkage analysis and then subsequent single gene sequencing in or from affected DNA region. For example, in family with the dominant type of inheritance and cysts was very often analyzed PKD1 gene, which is responsible or associated with polycystosis of kidney. In these days, the routine genetic diagnostic lab use clinical gene panel that allows to analyze 200 to 300 genes that are associated with kidney diseases, or they can use whole exome sequencing that allows to analyze all parts of DNA that encoded proteins, which is around 20,000 genes, but only 2% of entire DNA. And there is another approach which we often use, and it's analysis of whole genome sequencing. That is analysis of entire DNA molecule. And there are three types of responses from routine genetic lab or genetic diagnostic lab that the nephrologist can obtain after the testing. The first type of response is positive, is the best for patient because the pathogenic variant was identified and the genetic diagnosis can be made. And if possible or if available, nephrologist can start with the specific treatment. The second type of response or result is inconclusive. It means that suspicious variant in a known gene to be associated with kidney disease was identified, but there is no biological evidence for this specific variant and disease development. These variants are known as variants of unknown significance, and it needs our additional experimental tests to close the diagnosis. The worst case scenario is negative result. It means that no gene variants meet the criteria of type of inheritance, type of disease, and population frequency was identified. In rare genetic kidney diseases, the most often result from the routine diagnostic lab is either inconclusive or negative result. And these cases are cases for our team, for our lab. So now I will show you two examples of how knowledge of biology can help in accurate genetic diagnosis. First case is a case of Australian family that clinically fits to tubulointerstitial kidney disease. The proband in the family pedigree in the red square was analyzed by the routine genetic testing club, and one of the reported variant was UMOT variant. But this variant that affects structure of the uromodulin at the position 55, position amino acid. But this variant was classified by the bioinformatics as a benign, which means not pathogenic, not related to the disease. But the nephrologist contacted Dr. Blair with the question if this variant could be pathogenic for the disease. And Tony Blair referred this family to us. So what we know about uromodulin? Uromodulin is produced by a very tiny part of kidney by a specific cells that called tubular cells, and these cells processed uromodulin, which is exposed on surface of tubular cells, some of the tubular cells, and from where it's released into the urine. However, mutated uromodulin that cause ADTKD is not properly processed by the tubular cells and gets stuck inside of the cells, is not able to reach membrane of the cells and is not released into the urine. So we started with the analysis of uromodulin in kidney biopsy from patient with investigated variant, and we found that uromodulin is retained inside of tubular cells and is not present on plasma membrane, which corresponds with absence of uromodulin in the patient's kidney, in the patient's urine. So we concluded that formerly benign variant, we reclassified this variant for pathogenic based on the results that UMOT is retained in the kidney and absent in the patient urine. So we solved the case of this family as ADTKD due to UMOT mutation, and this family is now included in the registry of Dr. Blair. So the second example or second case which I would like to show you is a family with, again, ADTKD, autosomal dominant tubulointerstitial kidney disease, which was referred to us directly from Dr. Blair and Kendra Kidd. In this family, the routine testing club, genetic testing using clinical gene panel was negative. And Professor Blair and Kendra Kidd sent to us the samples. And by the whole exome sequencing and whole genome sequencing that we did, we identified variant in ApoA4 gene that have never been associated with development of tubulointerstitial kidney disease before. And the staining of ApoA4 in a kidney biopsy from patient from this family was not informative. So we didn't know if this variant could be pathogenic, could be cause of the disease, but within next 2 years, through the registry of Professor Blair and Kendra Kidd, we identified additional 4 families with another variant in ApoA4. But we still couldn't find the biological mechanism why in these families is developed chronic kidney disease and kidney failure until one of the patients was subjected to nephrectomy and we analyzed whole kidney uh, tissue, and we found or detected the pathogenic protein structure in a very deep part of kidney, which is not typically included in kidney biopsy. And these structures, pathogenic protein structures called amyloid, and these structures are formed mainly by mutated ApoA4 protein. So we can conclude that in these families, we identified a novel genetic cause of ADTKD, autosomal dominant tubular interstitial kidney disease, and we also identified the biological mechanism of the failure and kidney damage in these families. So by our approach, which is a combination of genetic testing and the identification of biological consequences and molecular mechanisms of the disease development, we significantly contribute to building of the registry with TKD cases of Professor Blair and Kendra Kidd. In this registry still remains around 70 families with ADTKD and unknown etiology. And we are testing 6 novel genes potentially associated with this disease. So we can conclude that genetic cause of rare hereditary chronic kidney disease is solved by routine lab testing from around 40%. Cases with inconclusive or negative results can be solved by clinical research team as we are. And knowledge of the basic biology of causal proteins is an important step to solve genetic diagnosis. Accurate genetic diagnosis of the disease is important for genetic counseling, prenatal diagnosis, selection of a healthy kidney donor within the family, and therapy development. So thank you for your attention. And my thanks also go to my colleagues from Research Unit for Rare Diseases in Prague, and also to the team of Tony Blair and Kendra Kidd from Wake Forest School of Medicine. So thank you for your attention.
Thank you so much, Martina. That was so interesting from what I could pick up anyway. But to see the extent of and your process of how you go detective work to figure
Yes.
out when there's mysteries and the what's available in the research lab to dig deeper, all in the effort to understand this disease, find out the biological pathway, and ultimately find a treatment. So thank you so much for you and your colleagues for all the work that you're doing.
Mhm.